Right, standing desk. Let's try this. So today's topic is metabolomics. Yeah, which is a neat thing that you can do. And as with everything worth doing, there's a whole lot of bioinformatics underneath it. So the metabolome is all the metabolites that the cells of an organism are making. What's a metabolite? Anything that is on the way to basically be produced or be broken down is potentially a metabolite. Just sort of evidence that this thing is living. So, like, think nucleotides, that's like a finished metabolite. But the nucleoside without the phosphates, that would be a sort of on the way to producing the nucleotide. And then there's all the other versions of this. So you got enzyme after enzyme, like an assembly line, putting together each of the parts that makes a little molecule of life. And so each one of those little byproducts, the little intermediate stages, those are all metabolites. And yeah, you can read these. For the most part, the technique is going to involve mass spectrometry, just like we talked about in doing protein structure, only you're going to be throwing little molecules with your laser at the detector instead of throwing chunks of a protein. Yeah. Otherwise, pretty much the same thing. All right. Why? Oh, yeah. Because you can tell a lot about what an organism is doing by what its metabolism is showing you. Because metabolism relates to the enzymes that make those products. And so you can tell how much of a certain enzyme is turned on. And if you have a lot of metabolite, it kind of implies that all the enzymes leading up to that, that entire pathway are activated. Yeah. And if it's an intermediate metabolite, if it's like halfway through the process of becoming something else and you have a whole bunch of it, then it might also mean that the next enzyme that would turn it into the next thing, maybe that one is more or less switched off right now. So in addition to your genetics, what sorts of enzymes you have, those are not going to vary very much from one person to another. There's also going to be the epigenetic information. So epigenetic is what we call information that is not encoded in your genome, but still produces a phenotype, some kind of biological, meaningful difference, potentially meaningful, anyway. And so there are epigenetic phenotypes. Like, for example, weirdly, things like trauma can have effects on the cells. Like, going through a bad experience does not change your DNA, but it will change the amount of certain enzymes that you have in certain cells, and that'll change the way that they respond to outside stresses. And so, yeah, you get these phenomena like if your grandmother had a hard time during the Depression, finding food, then there is an epigenetic signature that actually gets passed down over a couple of generations at least. And so, yeah, these things, for better or worse, can go on. And they don't involve changes to the DNA, but they involve changes to the way the DNA is used and basically, yeah, the amount of things that are made from the DNA. And those actually have a meaning. So, yeah, let's get into it. So the idea is that you have DNA that makes RNA, RNA makes proteins, and the proteins make all the other stuff that keeps you alive. That's pretty easy, right? Yeah. A lot of these are. And I love this little dancing animated GIF of the stomach is about to metabolize all that good food. Look how happy that stomach looks. It's got his little workout stuff on. Oh my gosh. Yeah. You go, stomach. You go. So, on the Y metabolomics side, yeah, so a little bit of DNA can produce a large number of RNA molecules. Those RNA molecules can then produce more polypeptides, but maybe you have to activate the polypeptides. And like if you're just doing mass spectrometry on the proteins, like to see the proteome, which proteins are being made, the problem with that is that it's usually got to be destructive. So you need to take some cells and dissolve them and get out the proteins and, throw them at the wall with your mass spec machine. Whereas with a metabolome, you can sometimes do it from a little drop of blood or just some sort of secretion, something like that. And so you can potentially find out what's going on inside the organism without dissolving the organism. One way they do this, and they probably did it on you when you were a baby. If you ever see a baby born, one thing they'll do is they'll prick the heel of the baby, get a little tiny drop of blood out, and put it on a little bit of paper or in a little tube and send it off to the lab. And they're going to do metabolomics on that baby blood to check if things like phenylketonuria, like various conditions that are treatable, are there, because then you can start treating them right from the beginning. Because if you wait for something to kind of go wrong and then say, oh, the cause was, genetic or epigenetic, then by that point you have a problem. So you catch it early, you can treat it early, and a person can lead more or less a normal life. But everything goes into the metabolome. So it's not just the proteins you make, it's how many of those proteins are going to be activated or at a particular site where they can, interact with a substrate. It's going to be what other chemicals are present that are coming in from the environment. I don't know why they're showing Viagra and beer here, but apparently that's what this person thinks you guys are up to. Yeah, which I would have thought that would skew a little older, at least the Viagra, but who knows? I don't know. And then they're saying that exercise, and I'm assuming that's a picture of exercise down there, can also definitely affect this. And so, at least in theory, you ought to be able to detect exactly what's going on. If you can decode the metabolome, decoding the metabolome, that's going to take a whole lot of bioinformatics to do it well anyway. So you got genes which make enzymes and enzymes which make metabolites, and you can screen the metabolites, which kind of tells you which enzymes are turned on, which in turn can kind of give you a clue as to what's going on with the genes. And so you can completely non-destructively get some sort of biological information. And it's information on the most important parts, parts that are enzymes that are actually contributing to keeping this organism alive and making it do whatever the heck it does. So you would have different metabolomes for different types of cells, for example. Neuron metabolome is going to look nothing like a gastric, cell metabolome, and that's okay. It really shows you how the same DNA in all your different cells can be put to work in different ways. All right. Why is it hard? Well, with nucleotides, there are only, it's got 5 bases here. That's A, C, G, T, and U if we're talking about RNA. But really, T kind of is U, so, more or less 4 of them. 20 amino acids, 20 common ones. I don't know, maybe 2 other less common ones, really less common ones. And you get a bunch of modified nucleotides and tRNAs that they're pretending don't exist here. But whatever, it's only science, right? We just dabble in precision here. But in terms of metabolites, you can have 10 to the 5th different molecules that a cell is actively making, breaking down, that are just like intermediate steps. It's like if you wanted to see what's happening in a factory, but the factory and the machines in it are somehow invisible, but you could just watch everything that's going down the assembly line and count how many of each part there are. You could tell what kind of factory it is. You could tell which parts are running well, which parts are sort of building up, a pile of excess parts. What are the rate limiting steps? All this stuff. It really adds a layer of understanding to how life works. Why is it difficult? Well, you're dealing with itty bitty little chemicals, and a lot of these are labile, Labile. You know that word, right? Labile means a thing that's going to break down, maybe even without you doing anything. So like a lot of compounds will react, given enough time, with water. And cells are pretty watery, in case you haven't heard. So some of these, if you do not actively go in there and freeze everything or purify everything within minutes or seconds, then the metabolome will change. At least some of the chemicals will go away or turn into something else, and you may get a false read on what's actually going on. So there are also giant differences in molecular weight. Little 20 Dalton things might be interesting, as will 2,000, 2 kilodalton giant molecules. And some of them are real common, some of them are real rare, and rarity doesn't mean it's less important. It just means you probably need less of it in order to do something. But like enzyme cofactors or something are totally essential for the activity of the enzyme, and you're not going to have a lot of them hanging around. But yeah, the ones that are there are going to do some work. So, okay, so metabolic profiling is like a quantitative study. So we're going to look at particular metabolites on a pathway, and we're going to try to find out the amount of each one of these things that's there. And we can do this with, like, imagine an animal that we don't know as much about. Imagine something like a sponge or a coral or something like that. So it's a living animal, but with a really different biology to you and I, and possibly one that hasn't been studied as much if you're talking about certain species of these things. You would be able to go into them and you'd still be able to do metabolomics, but you wouldn't necessarily know what all the things you're seeing are. You could just say, oh, there's a whole bunch of this. It looks similar to this compound, maybe if you added a methyl group onto it or something like that. And this can be the start where you start to get a handle on how these organisms work and how they're, how they're tweaking the general sort of molecules of life in order to get by. Fingerprinting is going to attempt to just get like the entire array, like what are all the different metabolites that you're making. It's not really going for quantity, it's just going for what is the full array of what's going on here. So with the quantitative stuff, that might tell you sort of subtle differences between one cell type and another one. Fingerprinting can tell you whether you have a virus infection, whether you have cancer going on. You'll have generally different metabolites made or potentially different amounts of metabolites made. So these two are pretty much linked. And good studies can actually do a decent job on both of them, which is very nice. But in general, you're going to be aiming for more diversity or more quantitativeness. Yeah, quantitation, I guess, is how we would say that in English. Okay, so let's pause right there, and then we'll do the second part. Yeah, look at how does metabolomics work? Yeah, short answer, mass spec. Long answer, stay tuned.